Good afternoon, children. Welcome to the class. So, now today we'll take up the next subtopic of our chapter human reproduction. Next, what we have to take up is the fertilization. So, the fusion of a haploid male gamete, that is a spermatozoon, and a haploid female gamete, that is the ovum, to form a diploid cell, the zygote, is known as fertilization. Now, where does this fertilization occur? That is, what is the site of fertilization? Fertilization in human female is internal, like other mammals. It takes place usually in the ampulla of the fallopian tube. Previously, it was said that it is in the ampullary isthmus junction, but actually, now what is known is the latest what is known as it is in the ampulla of the fallopian tube. Now we will take up all these points or headings one by one, which are displayed on your screens. Now this the first is the arrival of the spermatozoa. So after insemination from the vagina, the sperms they reach the ampulla partly by their own effort and partly by the action of the uterus. Now, what are the sperm's efforts? The sperm present in the semen travel a long way through the uterus into the fallopian tube by swimming in the fluid medium. The sperms may swim at the rate of 1.5 to 3 mm per minute. A single ejaculate of the semen contains about 200 to 400 million sperms in around 3 ml of the fluid secretions. But only 100 or so sperms reach the fallopian tube. Now, what is the role of uterus in sperm transportation? Certain activities of the female reproductive tract help the sperms reach the site of fertilization. For example, extension of the cervix into the vagina aspirates the semen into the uterus from the vagina, and contractions of the uterus and fallopian tubes propel the sperms upward. Sperms may reach fallopian tube within 5 minutes. Although a sperm cell can survive in the female reproductive tract for about 1 to 3 days, it can fertilize the oocyte in about 12 to 24 hours following ovulation. Now, as far as the next event is concerned, that is the arrival of the egg. The egg released from the mature Graafian follicle of the ovary. That is the process of ovulation, which we studied yesterday, is received by the nearby fallopian funnel and sent into the fallopian tube by movements of fimbriae and their cilia. It is in the secondary oocyte stage with the second meiotic division in progress. The oocyte can live for 72 hours but can be fertilized only during the first 24 hours of this period. Now, what do the recent research says? It says that this period is only for 6 hours, according to a latest study. If copulation occurs at the appropriate time, the egg is surrounded by numerous sperms. Most of the sperms contribute enzymes which disrupt the follicular cells, still investing the egg. Only one sperm succeeds in fertilizing the egg, and others degenerate and are engulfed by the leukocytes. Now, next is the capacitation of the sperms. Now, what does this mean? The sperms in the female's genital tract are made capable of fertilizing the egg by secretions of the female genital tract. These secretions alter certain molecules deposited on the surface of the spermatozoa from the semen. This process is known as capacitization of sperms. It takes around 6 hours. The secretions of seminal vesicles, prostate gland, and Cooper's gland present in the semen contain nutrients which activate the sperms and substances that neutralize the acidity in the vagina. Sperms need an alkaline medium in order to be motile. So these changes occur. Then next is the chemical and physical events of fertilization. Now here this event, it comprises of 5 total processes, that is the acrosome reaction, the cortical reaction, the sperm entry, karyogamy, and activation of the egg. Here you can see them on your screens. So the first is acrosome reaction. The secondary oocyte reaches the fallopian tube and is surrounded by zona pellucida and corona radiata. A capacitated sperm passes through the corona radiata to reach the zona pellucida. One of the 3 glycoproteins of zona pellucida, named ZP3, functions as a sperm receptor and binds to the complementary molecule on the surface of the sperm head. Binding of the sperm head to the receptor molecule ZP3 induces the acrosome of the sperm to release its hydrolytic enzymes by exocytosis. These enzymes are collectively known as sperm lysins. Main sperm lysins include hyaluronidase that hydrolyzes hyaluronic acid of the follicular cells, corona penetrating enzyme. It dissolves corona radiata portion around the secondary oocyte by hydrolyzing their ground substances, zona lysine or acrosin. It helps to digest zona pellucida. All these enzymes dissolve the corona radiata and the zona pellucida and enable the sperm to reach the plasma membrane of the egg. The above changes in the sperm head are called acrosome reaction. Right? So all these changes, they are known as acrosome reaction. Calcium ions play an important role in the acrosome reaction. Other essential parameters are like proper pH, proper magnesium ion concentration, and temperature. Acrosome reaction also exposes some proteins in the sperm membrane that binds to receptors on the egg plasma membrane. Now the plasma membrane of the sperm fuses with the egg plasma membrane, making it possible for the sperm contents to enter the egg. All these things you can see in this particular diagram, which shows the different phases of the sperm entry here. Binding of the sperm to the egg induces depolarization of the egg plasma membrane. Depolarization causes fast block to polyspermy that checks additional sperms from joining the egg's plasma membrane. Then the cortical reaction. Immediately after the fusion of sperm and egg's plasma membrane, the egg shows a cortical reaction to further check the entry of more sperms. In this reaction, the cortical granules present beneath the egg's plasma membrane fuse with the plasma membrane and release their contents, that is the enzymes between the plasma membrane and the zona pellucida. These enzymes harden the zona pellucida, which functions as a sure block to polyembryony. Next is the sperm entry. The egg extends around the entering sperm finger-like processes which are known as microvilli, which constitute a fertilization cone. This fertilization cone takes up entire sperm into the egg. The distal centriole of the sperm divides and forms two centrioles to generate the mitotic spindle for cell division. The mammalian egg lacks centrioles of its own, You know that this uh, secondary oocyte stage, the second meiosis is restricted to the second meiosis is restricted at the uh, metaphase 2. Next is the karyogamy. So the sperm entry stimulates the egg to resume and complete the suspended meiosis 2. This produces a haploid mature ovum and a second polar body. At this time, the head of the spermatozoon, which consists of the nucleus, separates from the middle piece and tail and becomes the male pronucleus. The second polar body immediately degenerates, and so does the sperm tail. The chromosomes of the ovum are not recognized, they are not organized into a proper nucleus. They represent the female pronucleus. The chromosomes of the sperm are set free by breakdown of the nuclear envelope. So the two sets of chromosomes go over a spindle formed between the centrioles. Mixing up of the chromosomes of both the spermatozoon and the ovum takes place, and this is what is known as karyogamy or amphimixis. This completes the act of fertilization. It combines the genetic information of both the parents. The ovum is now a diploid cell having 23 pairs of chromosomes, and so it is now known as a zygote. Zygon means they are yoked together. So the first cell of the new individual is the zygote. It has 46 chromosomes rather than an organized nucleus with a nuclear membrane. The mother is now said to be pregnant. No nuclear envelope is formed around the chromosomes as they are already double, each consisting of 2 chromatids, and the zygote is ready for the first cleavage. Now the last step is the activation of the egg. Sperm entry stimulates metabolism in the zygote. Within minutes, the rate of cellular respiration and protein synthesis increases. Fertilization is irreversible process and is mainly species specific. Sperm fertilizes an egg within 30 minutes of ejaculation. If egg is not fertilized, it disintegrates and it's absorbed by the leukocytes. So in the diagram displayed on your screens, you can see that different events occurring, that is the sperm passing through the corona radiata. Then you can see the lysing enzymes being released, then sperm passing through the pellucida layer and reaching the oocyte, then sperm and egg plasma membranes fusing together and enabling the sperm to enter into the egg, and finally the cortical reaction which releases enzymes to harden the zona pellucida in order to prevent the polyspermy. Now in this diagram you see the structure of the ovum at the time of fertilization. You can see the outer cells of the corona radiata, then the zona pellucida, then you can see the ooplasm or the plasma, this and the plasma membrane or the ulema. So you can see all these things plus the first polar body and the spindle which is formed for the meiosis II. So it is arrested here. The division is held here till the fertilization is effected. It is completed after the fertilization. Is that clear? Now, what determines the sex of the offspring? You know, in humans, each diploid cell has 46 chromosomes. Of these, there are only one pair of the sex chromosomes, rest are all autosomes. A human female has homomorphic sex chromosomes, that is X and X, they are represented by X and X, whereas the human male has heteromorphic sex chromosomes, that is X and Y. So this means that the female produces the same type of gametes, that is the each gamete having an X chromosome, whereas on the other hand the male produces two types of gametes, 50% having X chromosome and 50% having Y chromosome. So the sex of the child is determined at the time of fertilization when the male and the female gametes fuse to form the zygote. So what determines it? If the sperm which is carrying X chromosome fertilizes an egg, then the resultant offspring will be female, whereas if the sperm carrying a Y chromosome fertilizes an egg, then the offspring will be a male. This is how the sex of the offspring is determined. Now, what is the significance of fertilization? It provides stimulus for the egg to complete its maturation. It activates the ovum to develop into a new individual by repeated mitotic divisions. It restores the diploid number of chromosomes in the zygote by adding the male's haploid set of chromosomes. It makes the egg more active metabolically. It combines characters of two parents, that is, it introduces the variations which make the offspring better equipped for the struggle for existence and contributes to the evolution of the race. It determines the sex of the young one to be developed from the zygote. Ulema's depolarization and hardening of zona pellucida resulting after sperm entry check the entry of additional sperms. Fertilization introduces centrioles which are missing in the ovum. Now, the last thing what we will discuss today is the species specificity in fertilization. You know, a glycoprotein, fertilizin, composed of monosaccharides and amino acids, is secreted by ovum. Penetrating sperm also has on its surface a protein substance which is known as anti-fertilizin. The interaction of fertilizin of an ovum with the anti-fertilizin of the sperm is species specific. Because of this species specific interaction, the sperms adhere to the egg, and that is why they fertilize the egg of the same species. Is that clear? Okay, students, then see you tomorrow at the same time. Good afternoon.